Hello, my name is Brian Duff. I'm Technical Lead for the PSC portfolio. Welcome to PSC Foundations Module 4, Induced Pluripotent Stem Cells. This module contains two sections. Section 1, what are induced pluripotent stem cells? And Section 2, steps to derive induced pluripotent stem cells. Sidebar sections are used to highlight applications and useful additional information related to the curriculum. After this module, you should be able to understand the general overview of the reprogramming process and describe considerations and necessary steps for reprogramming. Section 1, what are induced pluripotent stem cells? All cells that exist in the body, both somatic and germ cells, originate from the pluripotent cells of the embryo. It was once hypothesized that mammalian cellular development only progressed down a path that becomes increasingly cell fate restricted, and that cells were not capable of bypassing these barriers. The recently developed induced pluripotent stem cell, or iPSC technology, which enables the induction of pluripotency in mature somatic cells by treating them with defined transcription factors, has created new avenues in basic research, disease modeling, and regenerative medicine. Dr. Shinya Yamanaka's remarkable study demonstrated that the oocyte can completely reverse the acquired memories of somatic cells. Before his breakthrough, differentiation had long been considered to be a one-way street, with traffic flowing from an immature state, a stem cell, to a more mature differentiated state. However, a small set of reprogramming factors can revert lineage-committed differentiated cells to pluripotency. In parallel, it was previously believed that unnecessary genetic information was deleted or terminally inactivated in cells committed to a specific state. This was a theory known as Weissman's barrier. Nuclear reprogramming demonstrated that not only somatic cells retain all genetic information, but they can also be rejuvenated by artificial manipulations to again acquire pluripotency. Shinya Yamanaka and colleagues developed mouse iPSCs in 2006 through a different method of reprogramming. Here they use a retrovirus to deliver into somatic cells, in this case mouse fibroblasts, a combination of 4 reprogramming transcription factors comprised of OCT34, SOX2, KLF4, and c-MYC, nicknamed the OSKM factors. A year later, in 2007, Yamanaka and his team applied the same reprogramming method for adult human fibroblasts to generate human-induced pluripotent stem cells. James Thompson's group reported the generation of the same human-induced pluripotent stem cells, though in this case they used a different delivery system, the lentivirus, and a different set of 4 factors, OCT3 and 4, SOX2, NANOG, and LIN28. For their revolutionary discoveries, Shinya Yamanaka and John B. Gurdon were awarded the 2012 Nobel Prize in Physiology and Medicine. Section 2, Steps to derive induced pluripotent stem cells. There are several steps in generating induced pluripotent stem cells from somatic cells. Firstly, one needs to choose the somatic cell type as starting cells for reprogramming. After choosing starting cells, reprogramming factors should be chosen, and the delivery method for transfection as well as culturing conditions should also be considered. These are the strategic choices prior to reprogramming. Reprogramming of somatic cells will begin with transfecting them with the reprogramming factors, also known as the pluripotency factors or the Yamanaka factors. After a few weeks of culture, a small fraction of transfected cells form colonies and become morphologically similar to embryonic stem cells. Once the fully reprogrammed iPSC colonies are generated, they will be picked and plated into maintenance media. The culture is then tested for pluripotency, markers of undifferentiation, and karyotyped. It is important to note that the entire reprogramming process is inefficient, time-consuming, and labor-intensive. Depending on the purpose of the iPSCs, be it cell therapy or research, several choices have to be made concerning the somatic cell type, the reprogramming factors used, delivery method, and culturing conditions. These parameters influence the reprogramming process, the efficiency, the cost, and the time of reprogramming. This has been described in detail in an article by Amol and Fukuya in 2018. Along with choosing the right reprogramming factors and delivery method for the generation of iPSCs, it's also important to use a source of cells that will generate the desirable cell types for further study purposes. Somatic cells from a variety of tissue types and functions have been successfully reprogrammed to iPSCs. The choice of cells for reprogramming depends on several factors, such as accessibility, cost, field of study, the therapeutic or disease relevance, and the ease of reprogramming. iPSCs were first established from primary mouse fibroblasts. However, several different types of cells have been used to produce iPSCs, including fibroblasts, neural progenitor cells, blood cells, keratinocytes, adipose stem cells, and more recently, urine-derived cells. The efficiency of cell reprogramming varies among different cell types. Reprogramming efficiency is also dependent on the donor health and the primary cell passage number. The following figure from Gonzalez and colleagues in 2011 describes two criteria for choosing a specific starting cell type for reprogramming: availability and ease of reprogramming. Availability refers to how easily the cells can be obtained without using invasive procedures. Ease of reprogramming was determined by reprogramming efficiency and/or a decrease in factor requirement. Some cell types endogenously express reprogramming factors and are more easily reprogrammable. For example, CD133 positive cord blood cells already express higher levels of KLF4 and c-Myc compared to fibroblasts. Therefore, CD133 positive cord blood cells can be reprogrammed to iPSCs without exogenous KLF4 and c-Myc. Both Shinya Yamanaka and James Thompson's group determined that only a handful of factors are needed to reprogram adult fibroblasts to iPSCs. The conventional OSKM cocktail by Yamanaka's group has been used extensively by research on a wide range of human somatic cells and delivery systems. Thompson's group provided an alternative combination of four factors: OCT3 and 4, SOX2, NANOG, and Lin28, known as OSNL. The factors that were used to reprogram adult cells are generally found in naturally pluripotent stem cells and are generally transcription factors. However, Lin28 is a microRNA binding protein. The OSKM and OSNL reprogramming cocktails have worked well on a wide range of delivery systems, albeit at low efficiency. Consequently, researchers have sought to discover new molecules that will enhance the reprogramming technique and improve its efficiency. On this table, we summarize some of them. Many of the transcription factors used for reprogramming somatic cells are part of a core pluripotency circuitry. These factors are pluripotency-associated genes expressed early during embryonic development and are involved in the maintenance of pluripotency and self-renewal. The expression of other pluripotency-associated genes, along with the minimal pluripotency factors, OSKM, can enhance the reprogramming efficiency or even replace some of the reprogramming factors. Non-coding RNAs and their modulators like LET7 are involved in the regulation of expression of core transcriptional factors. LET7 acts as a barrier by blocking the activation of pluripotency factors c-Myc, Lin28, and Sol4. The reprogramming of somatic cells into iPSCs is characterized by epigenetic changes, from DNA methylation to histone modifications. For example, histone deacetylase or HDAC inhibitors such as SAHA and valproic acid enhance reprogramming efficiency in MEFs. Valproic acid also promotes somatic cell reprogramming with OCT4 and SOX2 alone. As cells move towards pluripotency, somatic cells also gain the ability to proliferate indefinitely. Not surprisingly, two of the minimal pluripotency factors, c-Myc and KLF4, are oncogenes that enhance cellular proliferation. The p53 tumor suppressor protein promotes senescence and inhibits growth, thus having an inhibitory effect on iPSC generation. Overexpression of p53 inhibitor proteins, such as REM2 and MDM2, increase the reprogramming efficiency. Small molecules also improve cellular reprogramming efficiency, in part by promoting the activity of epigenetics, cell cycle regulation, and signaling pathways. In order to increase the amount of pluripotency factors present in an adult cell, these factors can be delivered into the cell through several methods. These methods can be characterized by several criteria, e.g., integrating or non-integrating, viral, non-viral, DNA and non-DNA methods. By integrating, we mean that the foreign genetic material is incorporated into the host cell genome. By viral method, we mean that the foreign genetic material is delivered into the host cells with viruses. Integrating viral systems were the first to be used to deliver transcription factors to generate iPSCs, but they have the disadvantage of incorporating their genetic material and contributing to teratoma formation. By avoiding integration, novel methods such as using non-integrating vectors, self-excising vectors, and non-integrating non-viral vectors represent iterative improvements upon this initial methodology. Such approaches provide significant advances in the safety and efficacy of iPSCs, which may then be applied for downstream scientific and clinical applications. Dr. Yamanaka's original work used an integrative viral approach to reprogram mouse and human fibroblasts. However, integrative viral approaches are considered less safe than other approaches. As iPSC derivation moves more and more towards the clinic, the safety associated with a particular derivation approach is increasingly scrutinized. The delivery of the OSKM transcription factors into mouse or human fibroblasts was originally achieved using MMLV-derived retroviruses and also lentiviruses. Lentiviral delivery vectors have also been successfully used to express different sets of reprogramming factors in somatic cells. Although they are efficient and reproducible, reprogramming using viruses entails the production of potentially harmful viral particles that express potent oncogenes. iPSC lines generated using these vectors carry randomly distributed viral transgene insertions, which could disrupt the expression of tumor suppressor genes if there are insertions in the open reading frames, or alter the expression of oncogenes if inserted nearby. The use of Cre-deletable or inducible lentiviruses has solved some of these problems, but viral systems still lack the safety required for therapeutic applications. If aiming to avoid the use of viral vectors, standard DNA transfection using liposomes or electroporation is a good alternative. Compared with viruses, however, transfection efficiency is much lower. To enhance the stable integration of non-viral constructs, researchers have moved to viruses based on the piggyback or PB transposon. The piggyback transposon is capable of excising itself precisely without leaving any footprint or remnant of exogenous DNA in the reprogrammed cell genome. Another non-integrating viral vector that has been successfully used for iPSC generation is the Sendai virus. These are very efficient in transferring genes in the form of negative strand single-stranded RNA into a wide range of somatic cells. Although they are very effective, the viral vector's RNA replicase is very sensitive to the transgene sequence content. Additionally, because they replicate, these vectors may be difficult to eliminate from the somatic cells. RNAs can be directly delivered into somatic cells by transfection as synthetic modified mRNA. These modified mRNAs can increase RNA half-life and translation efficiency. However, a fraction of the synthetic RNA can induce innate immune responses. To reduce this response, synthetic RNAs may be treated with a phosphatase prior to transfection. Addition of interferon inhibitors to culturing media helps to reduce the immunogenicity also. Finally, protein transduction is the first reported strategy to entirely replace gene delivery, albeit at attenuated efficiencies. Transfection refers to the introduction of genetic material, DNA or RNA, or a protein into a cell. In this case, the pluripotency factors are being introduced so that they will be expressed within a cell to instigate the reprogramming process. Once the somatic cells are transfected or transduced with reprogramming factors, the time frame for obtaining an initial reprogrammed colony can vary from 1 week to 1 month. In addition, this may require frequent reprogramming factor replenishing, for example, with protein-based strategies. Whereas self-replicating vectors such as self-replicating RNA only require a single transfection. After transfection has taken place, cells are slowly reprogrammed to pluripotent stem cells. This can take several weeks. The reprogrammed cells will expand in culture, undergo mesenchymal epithelial transition, and form colonies. Both the cell and the colony begin to take on distinct morphology of pluripotent stem cells. Reprogrammed cells need to be subcloned to ensure the clonal populations. For subcloning, each individual iPSC colony is isolated using a pipette tip or a pulled glass pipette or a fine needle. Then the iPSC colony is broken into smaller clumps of about 20 to 30 cells per clump and placed into maintenance culture medium. Once an iPSC line has been established, you would maintain the line with established methods. This may take several passages after the initial picking of the colonies. Finally, once the cell line is established, it is assessed and characterized for pluripotency, undifferentiated PSE markers, and the genotype. So in summary, we learned that human-induced pluripotent stem cells are pluripotent stem cells that are derived from non-pluripotent cells that have been reprogrammed to have embryonic stem cell-like properties. We also discussed the steps to derive induced pluripotent stem cells, and they included the choices to make prior to reprogramming around the starting cell type, vector to use, and reprogramming factors. Then we looked at how to reprogram non-pluripotent cells and how to culture and characterize newly reprogrammed iPSCs. Thank you for your attention.